Darwin's first postulate is that individuals vary. And we saw in class through the exercises that we did that this component is extremely important for making natural selection work. Darwin, of course, recognized that offspring tend to resemble their parents but do not exactly resemble them, and so he observed that there was variation among populations, but at the time the reason for that variation, the mechanisms behind that variation, were not well understood, and this is the problem that really kept Darwin awake at night. And the prevailing idea at that time, we've seen this slide before, was this idea of blending inheritance, that offspring were somehow some kind of average between the characteristics of their two parents. And we saw that this idea about the way inheritance works was problematic for the mechanism of natural selection because of this dilution effect. Every time a trait is averaged with another trait, it is only half strength. So this was a big problem for natural selection as a mechanism. But if we look at traits such as height and other continuously varying traits, we can kind of see why that explanation for inheritance might have made sense. We now know that these characters that seem to support blending inheritance, where offspring kind of seem to be an average of their two parents, we now know that although inheritance is discrete, gene expression can be extremely complicated, and there can be lots of different factors involved in the expression of these traits. A single trait can be influenced by multiple genes. That's usually the case in these continuously varying traits such as body height. Environment can have a huge influence. Things like nutritional status is going to have an impact on how tall you grow. And there can also be gene by environment interactions, and we'll see more about that as we get into the section of heritability in the course. So of course, much of this confusion was settled by the rediscovery of Gregor Mendel's work early in the 20th century, and both of Mendel's laws of inheritance really helped us to understand better how all of this works. So with the law of segregation, we now know that these traits are inherited intact. There's no blending or dilution of traits. A trait that disappears in one generation can reappear full strength in the next one, and that information is not lost through blending or mixing. The law that gives us the first clue to understanding why individuals vary, why offspring resemble their parents but not exactly, is this law of independent assortment. So each trait is inherited independently from other traits. And so if we look at this at the chromosome level, what independent assortment means is that during this process of meiosis, homologous chromosomes are separated and then duplicated, as we see here. And then in the two stages of meiosis, meiosis 1, there's the homologous pairs are separated into separate cells, and then the individual chromatids are then separated into 4 gametes after meiosis 2. So that means that these copies of the chromosomes that were inherited from the father and the ones that were inherited from the mother can recombine into any combination. So here, with just these 2 chromosomes, we've got blue with blue, or we can have blue with yellow, or we can have yellow with red, or we can have blue with red. And so this allows the different characteristics of the 2 parents to be scrambled in a new way in each individual gamete, and therefore in each individual offspring. So this gives us novel combinations of alleles. In addition, during meiosis, another kind of shuffling of the deck can happen in a phenomenon known as crossing over or recombination. So recombination happens in the early stages of meiosis where the 2 homologous chromosomes line up, and they can get very close to each other and actually fuse at some point and exchange the ends of the chromosomes. So here you see the blue one is crossing over the red one, and they can actually switch those ends. So now down here you see the chromosome that's mostly red has this segment from the blue chromosome. This one that's mostly blue has this segment from the red one. And so when we separate these finally into gametes, we've got one that's identical from the one that came from the father, one that is a mix of the two chromosomes with a little bit of the blue segment, another one that's mostly blue with a little bit of the red segment, and then another one that is identical to that paternal chromosome. And we see the exact same thing here replicated for this other gamete. So again, we've got the green and yellow crossing over. This can happen anywhere on the chromosome. It can exchange a lot of material or just a little bit. Here we've got just a little bit of this yellow chromosome recombining with the green one, and a little bit of the green recombining with the yellow to get two that are identical to the original chromosomes and two that are altered with different genetic material. And so this affects something known as linkage. So linkage is the tendency for alleles of different genes that are located on the same chromosome to be inherited together as a unit because they are on the same chromosome, and that chromosome is passed on into the gametes intact. And so when you have crossing over, an allele located here on this red chromosome, without the crossing over, it was linked to the allele at a separate gene nearby on the chromosome. And if that crossing over had not taken place, those two alleles of those two separate genes would have always been inherited together as a unit. But then the crossover broke them up and split them onto different chromosomes. And so now you've got a different combination of alleles on the same chromosome. So this is another way of sort of shuffling the deck and having different combinations of traits than what you found in the parents. So both recombination and independent assortment are ways of getting different combinations of traits. What about the emergence of entirely new traits? New traits are created by one thing and one thing only, and that is mutations. Okay, so we're going to call mutations the source of everything new under the sun. Everything else is just reshuffling of the deck. This is where you get new cards in the deck. So what are mutations? A mutation is any change to the genomic sequence. And so now that we have more information about the structure of DNA, we know something about the genetic code, we can now go a little bit beyond what Mendel knew and investigate how these mistakes can be made in the DNA sequence. So we know that the very structure of DNA with these 4 nucleotides that bind to each other in a very specific way ensures pretty reliable replication of the gene sequence. It's not going to be very prone to errors. But errors do happen, and most of the time they are corrected immediately. There's an army of enzymes whose job it is to proofread and check these DNA sequences as they're being replicated to make sure that these errors are not left in place. But this is not a foolproof system. Some of these errors do make it through and get replicated, and these are called mutations. So the simplest kind of mutation is a point mutation, and that's simply when a single nucleotide is changed to another nucleotide. So in this case, a change of G to C. And this is a change in the molecule itself. And so these point mutations can come in different varieties. Remember that we have purines, A and G, that can change from one to the other, and that's a transition. So when a purine changes to a purine, or a pyrimidine changes to a pyrimidine, those are both known as transitions. Or purines and pyrimidines can change into each other in these 4 different ways, and that's called a transversion. And again, this is a change in the actual molecular structure of these nucleotides. So when this happens, it can have a number of different results. The first possible result is nothing, and this is known as a silent mutation. Certain changes in nucleotide sequence will lead to the formation of the exact same amino acid in translation, and so the protein would not be changed at all. So why is that? This is because of what we call redundancy in the genetic code. What this chart shows is actually the genetic code. So you'll recall that nucleotides come in these groups of 3 called codons, and each codon codes for a specific amino acid. And the way we read this chart is these rows in the grid refer to the first base in the codon, the columns refer to the second base, and each of these rows within a box refers to the third base. So in the example that we just saw, where we had a codon that was AAG turned into a codon that was AAA, both AAG here, so there's the G, code for lysine. AAA also codes for lysine. So that change in the nucleotide sequence does not affect the amino acid that's coded for. It's not going to change anything. And you'll notice that there is a lot of duplication of the same amino acid for different codons. So, for example, leucine is coded by UUA, UUG, CUA, CUC, CUA, and CUG. So there are 6 different codons that code for leucine. The other thing to notice here is that a lot of this replication is within the same box. So this box has all leucine, this one has all proline, and then you see this in several places. So what that means is that these point mutations are most likely to be silent if it affects the 3rd base in the codon. So for example, with G and C in the 1st position, no matter what's in that 3rd position, you're going to get alanine. So this is redundancy in the genetic code. Multiple codons specify the same amino acid. So what that means is if you get this silent mutation, you've got no problem. It's coding for the exact same protein, although you can detect this as a different allele using molecular methods. There's not going to be any sort of difference in the phenotype. Second, you can get what are called nonsense mutations. What that means is that one of the codons in the sequence is changed so that it produces a stop codon. So a stop codon tells the translation process to stop there. That's the end of the protein. And so this happens prematurely in cases of nonsense mutations. So what you get is an incomplete protein. It stops before it's finished. And it's likely to result in either a complete loss of function or at least a change in the function of that protein that's produced. Finally, you can have a category of mutations called missense mutations. And so what missense mutations mean is that there is a change in the protein that's coded for by that point mutation. And missense mutations can come in 2 varieties. One is conservative. So conservative means that there are groups of amino acids that have similar chemical properties, so similar binding affinities. So in this case, this purple color indicates an amino acid that's basically basic. And this has been substituted one basic amino acid for another basic. It's going to behave fairly similarly in the protein. And so it's likely to have fold into similar conformation because it'll have the same affinities for other molecules. It'll be not exactly the identical protein, but the chances are that it's probably going to work fine because it's similar enough to the original amino acid that it probably won't affect the protein that much. The other kind of missense mutation is non-conservative. And so this is when an amino acid with very different chemical properties is substituted. And so in this case, we've got a polar amino acid substituted for a basic one. And this is going to change something about the way the protein functions. It might cause it to fold its tertiary structure into the wrong conformation, so really change the binding properties of the final protein product, but it's going to result in basically the wrong protein that does not do its job. Probably is not going to work, or it'll work very differently from the original protein. It's not going to do the job that the original protein did. So we could see from that analysis that these point mutations can have anywhere from no effect to a very small effect to a very large effect. And this can be either because it completely changes a protein that's critical to some pathway. It can be a major change in a regulatory protein that is responsible for turning on and off other genes. And so this is an image from your textbook that I want you to just think about a little bit. So these are examples of the results of particular point mutations. So everything from albinism in this first image, some forms of polydactyly are the result of a single point mutation. So having 6 fingers instead of 5. cases of polydactyly combined with webbing of the fingers. Piebaldism is a disruption of the melanin expressing genes in sort of a patchy pattern on the skin. This next one is a very, very painful and debilitating condition called fibrodysplasia ossificans progressiva, in which some of the connective tissues in muscles and tendons and ligaments actually become ossified and turn to bone. So you can imagine that this would be an extremely debilitating condition, as well as some forms of progeria, which is an extreme acceleration of the aging process in children. All of these are the result of single point mutations that have just changed one nucleotide for another one. So these seemingly trivial changes can have very, very profound effects depending on what protein it is that they're changing and how they're changing it. So another kind of mutation is called an insertion and deletion. These are very closely related to each other, so I'm going to talk about them kind of together simultaneously. And so an insertion is when a small number of nucleotides are inserted into the genetic code. A deletion is when a small number of nucleotides is eliminated from the DNA strand. And this usually occurs as a result of misalignment between the two DNA strands during replication. So very common in DNA sequences that have a lot of repetition in the nucleotide sequence. Things can get misaligned as the two DNA strands come apart. So again, this disrupts the pattern of codons. You can have an additional amino acid inserted, you can have an amino acid removed. That's going to change the structure of the protein. Or if you have something that's not a multiple of 3, you can have what's known as a frame shift mutation, so that all of the codons downstream of that insertion or deletion all the codons will be altered from that point on, and we'll see in class how this happens. So these first 3 kinds of mutation that we've talked about result in new alleles. They're changes within the same gene that cause it to be an altered form of that gene. We can also have mutations that result in new genes, and this is most often caused by gene duplication, so simple copying of one or more genes. And this most often happens during this process that we talked about before called crossing over. So remember, this is when the 2 homologous chromosomes line up during meiosis and exchange part of the chromosome. But if these 2 chromosomes line up somewhat offset from each other, they're not exactly lined up, this crossing over is going to be unequal. So unequal crossing over is going to result in certain genes being duplicated and certain genes being deleted. So let's look at how this works. So misalignment causes a different number of genes to be exchanged. So if we look at these 2 that are not lined up, A should be here, right, B here, and so they're not lined up correctly, and then they come together and they touch and they exchange these ends of the chromosomes. Okay, so this one is not affected. It's not one of the chromatids it's crossing over, so we can see that this first one, number 1, still has A, B, C, D, E, F. It's completely normal. This one, the blue one, crossed over between B and C, but the orange one crossed over between C and D. So that means that one of these that resulted from this crossing over has lost C. Where did that C go? It went over to this other chromatid, because when it crossed over, it was not in the same spot. And so one of them got 2 copies of C, and the other one didn't get any. And again, this last one, number 4, is this one that was not involved in that crossing over. It has the normal complement, A, B, C, D, E, F. So what we see is a deletion in this chromatid number 2, C is gone, and a duplication in chromatid number 3. So remember, these are going to be sort of packaged during meiosis 2 into separate gametes. And so if C is something critical to survival, any zygotes that end up with chromatid number 2 are out of luck. They don't have C. They probably won't survive. If the organism can tolerate having 2 copies of chromosome C. The zygotes that form from these gametes are going to have a duplication. They're going to have 2 copies. If C is not critical, chromatid 2 can also survive in the population, and then we would see a deletion of that genetic material. So this unequal crossing over results in 2 normal chromatids, the ones that weren't involved in crossing over. One that's missing gene C, likely non-viable, and one with 2 copies of gene C. Probably fine, although it's possible that an overdose of the gene product might be harmful. So this is looking at the exact same process in a different way. Duplication by unequal crossing over. You can see a pair of homologous chromosomes during meiosis lining up incorrectly. So you can see this one's way over to the right, this dark green one. And so when these chromatids that are next to each other come into contact and exchange material, this light green one is basically not giving any part of the gene to the other chromatid. This dark green one is giving that entire gene to this light green one, and so the result is one of these chromatids has the entire gene sequence duplicated. This other chromatid has a deletion of that same gene. And so if we look, these pink boxes represent a promoter region and terminal region, and also these introns, intervening sequences. And when this duplication happens by unequal crossing over, this new gene still has its own promoter region, its own terminal region, it's got all of the introns intact. When we see this duplicated gene, it's going to have the exact same set of introns as the gene that it was duplicated from. And so that's one of the ways we can tell that this new gene was the result of a duplication event. And again, this deletion pretty much loses the entire gene as a whole. So this new gene can start to be transcribed immediately. It's got its own promoter, it's got all of the DNA sequences there that it needs to be transcribed. There's another mechanism by which genes can be duplicated called retroposition. What happens in retroposition is that the gene is transcribed, converted to an mRNA copy. That mRNA is then processed. The introns are spliced out and the exons are spliced together. Then an accident happens and this mRNA is converted back into a DNA copy by reverse transcription. Then that DNA copy can be inserted back into the genome anywhere and become part of the genome. Duplicate genes that are created by each of these mechanisms can be identified by their structure. So by duplication, you still have the identical set of introns as the duplicated gene, and this new gene is going to have its own promoter region that allows it to be transcribed immediately. This new gene that arose by retroposition, first of all, has no introns. They've been spliced out. Introns can't be used to show its homology with the gene it was duplicated from. It's not likely to be right next to the gene that it was copied from because it can be sort of spliced into the genome anywhere. And at the time it gets spliced back in, it's not necessarily going to have its own promoter region. It's possible for it to be spliced in somewhere in a region that has a promoter region for another gene, but either that has to happen or it needs to evolve a new promoter region for it ever to be transcribed. So these are very different mechanisms for having gene duplication that you can actually distinguish based on the structure of the genes themselves. Another type of mutation is an inversion. An inversion happens when there is a double splicing of the DNA strand. So it gets spliced here and here. That segment of DNA then kind of floats around, and it can actually turn itself completely around and reinsert itself back into the same spot, only in the reverse order. So here you've got purple on the left and green on the right, and this has just flipped itself over Now you've got green on the left and purple on the right, and the same sequence, only in reverse order. So what does this do? So basically you've got all of these genes that can be transcribed just fine in either direction. It doesn't make any difference in terms of the protein products, except possibly in these ones in the region of the break. If those were coding regions, those genes might be affected. But inversion basically disrupts this idea of linkage. So again, linkage is the tendency for alleles of different genes to assort together during meiosis, so they're connected on the same chromosome. We saw before that an allele located here is always going to be right next to an allele located here, but then this crossing over broke them up, and so now they're associated with other alleles that were on the other chromosome. But when there are the two homologous chromosomes, and one of them has an inversion and the other one doesn't, they cannot line up in this way during meiosis. The homologous genes are sort of looking for each other to bind together to allow them to line up in this way, and because the genes are now in a different order, they can't bond together and line up during meiosis. So this crossing over is impossible if you've got one chromosome with an inversion and the other one without one. So these inversions tend to cause the genes within the inversion to become locked together, always inherited together with that chromosome, and we call this a supergene, a whole bunch of genes that are inherited together as a unit. And this is fairly common in fruit flies. There's a lot of research showing how locking together beneficial sets of alleles can be a selective advantage in many cases. And so a lot of times these inversions are actually supported and maintained by natural selection. The last two kinds of mutation I want to talk about are chromosome fusion and genome duplication. So both of these two result in a change in the chromosome number. So chromosome fusion is exactly what it sounds like. Two chromosomes get stuck together, become fused together to form one chromosome. And this is extremely rare, but we do have some evidence of it happening. And one of them is from human chromosome 2. So chimps have 24 chromosome pairs. Humans, as you know, have 23. And if you compare the banding patterns of human chromosome 2, they match perfectly with the banding patterns of 2 chimp chromosomes. So here you have the actual karyotype image, and you can see the banding, and this is diagrammatic form. You can see how these bands on the chromosomes exactly line up between these two separate chimp chromosomes. Not only that, but as you'll recall from Gen Bio and genetics, that there are these structures called telomeres at the ends of chromosomes, and they're associated with particular nucleotide sequences. And those nucleotide sequences that are associated with telomeres are found right in the middle of human chromosome 2, right at the point where these two chromosomes might have joined together to form one chromosome. Pretty strong evidence that this one chromosome in humans, chromosome 2, was actually formed by chromosome fusion of these two chromosomes found in chimps and presumably found in the common ancestor of chimps and humans. So because this results in a change in chromosome number, this can allow very, very rapid speciation to occur, because in vertebrates especially, in most animals, you cannot mate with another organism with a different chromosome number. So this is a mechanism for very rapid speciation. Secondly, we have duplications of the whole genome. So this can happen in a couple of different phases of meiosis, either during meiosis 1, if the two homologous pairs don't separate properly, or during meiosis 2, if the two chromatids don't separate properly. You can get a doubling of the number of chromosomes in the gametes, and this can lead to genome duplication. This is actually much, much more common in plants because they're more likely to be able to self-pollinate, and so if they do have a doubling of chromosomes and become tetraploid rather than diploid, they can self-fertilize and so have a mate with the same chromosome number, and this is actually a very well-known mechanism for speciation in plants. It's extremely rare in animals, but there is some evidence that it has in fact happened on occasion. And one of those pieces of evidence are these Hox genes. And we mentioned Hox genes when we were talking about molecular homology and evidence that all organisms are joined together in this great tree of life and can be traced back to a common ancestor. Their gene sequences are similar enough that they can be definitively homologized between all animal groups. And so we saw that these Hox genes are present in Drosophila and mouse, that we have homologous sets of Hox genes between those two groups. And we also saw that mice have not one but 4 Hox clusters. So what are these Hox genes? Remember that these are regulatory proteins. They're responsible for turning other genes on and off, determining where in the body they're expressed. They're mainly involved in anterior to posterior body patterning, segment identity, so knowing which segment of the body should produce which structures. And we discovered these Hox genes because of these mutations called homeotic mutations. So homeotic mutations are mutations that involve the wrong body structure appearing in the wrong place. So one of these homeotic mutations is called antennapedia, in which legs grow where the antennae are supposed to be. So wrong information here about what body segment these legs are in. Another example is a homeotic mutation called bithorax. So in normal flies, there's one set of wings and then a very small set of halteres instead of the second set of wings characteristic of insects. And in this bithorax mutation, this first thoracic segment, which normally produces wings, is replicated, so wings are produced instead of halteres in the second thoracic segment. And so, and again, it's an error in location of the correct body segment. So one thing this reminds us of is this fact that every single cell in the body carries all the information that's needed to produce every structure in the whole organism, and what structures develop where simply depends on which genes are turned on and off. And so when these Hox genes are disrupted, we end up with these mutations that are errors of location. So there's very strong evidence that these individual Hox genes in Drosophila are the result of gene duplications due to unequal crossing over. They often have comparable introns. They've got their own promoter regions. There's good evidence that these arose all due to gene duplications. But then the best explanation for this whole scale replication of the entire Hox clusters is a whole genome duplication that happened twice in the gnathostomes. So all gnathostomes have at least 4 Hox clusters. So one whole genome duplication that led to the second set, another whole genome duplication that led to the other 2. So both of these 2 would have been replicated again in the second whole genome replication. And we can actually plot this onto a phylogeny and see exactly where these different things happened. So outgroup, we can consider this. Drosophila has 1 Hox cluster, so we can be sure that that is the plesiomorphic condition, the ancestral condition. Cephalochordates, which are close relatives of vertebrates, but they're not vertebrates, also have a single Hox cluster. Lampreys, which we saw are vertebrates but not gnathostomes. By the time lampreys branched off, there had been 1 whole genome duplication, so they have 2 Hox clusters, and somewhere on the line leading to gnathostomes or the jawed vertebrates, there was another duplication event that led to the 4 Hox clusters. So our discussion of all of these different types of mutation lends itself well to sort of a nice categorization of the different kinds of mutations. So we saw that point mutations and insertions and deletions lead to new alleles, so different forms of the same gene. Gene duplication and genome duplication both lead to an absolute increase in the number of genes. So gene duplication is just going to increase the number of genes by 1. Whole genome duplication is going to completely double the amount of genetic information there. And this is extremely important to evolution, as we will see and discuss in the next class. We saw that Chromosome fusion and genome duplication can result in very rapid speciation because it's essentially an instant change in the number of chromosomes. And then we saw that these inversions increase linkage and create supergenes, even though most likely they don't result in any kind of change in phenotype because the same genes are still present.